Hello everyone. Today we are going to learn about the lytic and lysogenic cycles. Viruses are very specific in nature to which host they have to affect, and in that particularly which cell in the host it has to infect. Even the smallest and simplest prokaryotic organisms like bacteria, they are also will be infected by the viruses. So the viruses which will infect the bacteria are known as bacteriophages. So when the bacteria are infected by the viruses, they used to show two different type of cycles. One is lytic and another one lysogenic cycle. So what is lytic phage? Lytic phage, these are the virulent viruses. When infect the bacteria, it will happen. So when lytic phages infect the bacteria respond by producing large number of viruses. At the end of the particular incubation period, then the host cell bursts out by releasing new bacteriophages to infect other host cells. So this cycle is known as lytic cycle. And another one is lysogenic or temperate phages. Here the bacterial cell will not burst. Here what will happen, the viral nucleic acid used to be integrated into the bacterial chromosome and it will replicate along with the bacterial chromosome. Sometimes there is a transition phase, that is some bacteriophages will exhibit from lysogenic to lytic phase. So how it will exhibit lytic phase means, after lysogenic phase, then the release of virions from the host cell, it will happen by either lysis or process by that is reverse phagocytosis. What is reverse phagocytosis? That is movement of the solid molecules to the outside. That is known as reverse phagocytosis. So here we can see in this picture the lytic cycle and lysogenic cycle, it will be differentiated into different stages. Here you can see in the first phase, that is infection of the bacteriophage, where it attached to the bacterial cell, and then after that release of viral nucleic acid into the bacterial cell. Here the bacterial this viral nucleic acid, it becomes circularized and it will start replicating within the bacteria. After proper replication, after incubation time, then it will encapsulate the nucleic acid, that is capsid molecules will be synthesized and it will incorporate the viral nucleic acid and new bacteriophages used to be formed. After formation of sufficient bacteriophages, the bacterial cell lysate and it will release the new bacteriophages, that is progeny bacteriophages, to infect the new bacterial cells. Whereas in the other phase, that is lysogenic cycle, here the viral nucleic acid will integrated into the bacterial chromosome and it used to be look like a separate gene, and it will replicate along with the bacterial chromosome and it will be transmitted to the progeny bacterial cells. So it is replicating along with the bacterial chromosome. Here you can see. Or otherwise, sometimes this lysogenic phase, some bacteriophages will induce the lytic phase from the lysogenic phase. So this lytic cycle will be can be studied into different stages. Let us see what are those different stages. Here let us see the replication of the bacterial viruses. First, the phage will attach to the bacteria by its tail fibers. I will explain the bacteriophage structure and different components in the upcoming class. Just today you remember these lytic and lysogenic cycles. So when the phage will attach to the bacteria by its tail fibers, what it will happen? Last class we discussed about the bacterial cell structure. So in that sheath is also used to be there. Then bacterial sheath used to be contract, driving the tail core into the bacterial cell wall and membrane. Then virus, it will start injecting its DNA, like how the syringe injects a vaccine, like that the virus, it will inject the DNA into the bacterial cell. Then the phage replicate inside the bacterial cell by utilizing host cell biosynthetic machinery. Even in the plant also, virus will do the same thing. It will utilize the host cell synthesizing machinery. Then after 25 minutes of the initial infection, nearly you have to remember this, how much capability it is having. 200 bacteriophages will be produced, and how they will come out? By breaking the bacterial cell, that is lysis of the bacterial cell. So the lytic cycle is divided into different stages, like first one is the adsorption stage, then penetration, then third one is the synthesis of the viral components, and fourth one is the assembly of the prosenivirions, and fifth one is the release from the infected cells. We will discuss each one in detail. Coming to the first step, that is adsorption phase, the name itself indicating that as the adsorption of the virus particles to the bacterial cell. It is the term adsorption, not absorption. Adsorption means surface attachment of the things. So here this is the first step in the lytic cycle, where the virus used to be attached to the bacterial cell by its tail fibers. Then the tip of the viral tail becomes attached to the cell via specific receptor sites. Specific receptor sites means these receptors are nothing but proteins which will recognize these bacteriophages, and bacteriophage will recognize the specific receptors present on the bacteria, then only it can capable of attaching to this bacteria. So you may get doubt that if specific receptors are absent on the bacteria, what will happen? Automatically the virus will not able to attach to the bacteria in the absence of this receptor sites. So when some mutations will happen in the bacteria, if it will lose it will lost this receptor sites, then it will automatically it will become resistant to this bacteriophages. And this attachment is also very specific in nature for the certain viruses, and susceptible bacteria have these complementary molecular configurations to their opposing receptor sites. So when it will become mutant, then it will be resistant to the bacteriophage attack. Coming to the second step, that is penetration. Penetration of the phage into the host cell. It is a mechanical force. Because of the mechanical force applied by the tail of this bacteriophage, then the viral nucleic acid, it will integrate, it will come into the bacterial cell. So this mechanical force, it will be facilitated by, how it will be facilitated means localized digestion of this bacterial cell surface with the help of enzymes that is released by the this bacteriophage tail, that is lysozyme, or the bacteriophage, it will induce the bacteria to produce degrading enzymes, that is viral activation of host degrading enzymes. Because of degrading enzymes or because of the lysozyme, the bacterial cell, it will disrupted, so that virus makes an entry of that nucleic acid into the bacterial cell. In the T1 phages, the penetration is achieved. These T1 phages, these are the virulent phages which will infect the E. coli, used to be present in the human intestine. So here how the penetration will be achieved means, the tail fibers of the virus, first they will attach to the bacterial cell, and it hold the tail firmly against the cell wall, that it will attach very strongly against this bacterial cell wall. Then bacteria sheath, it will contract, then driving the tail core into the cell wall and its membranes, that is the tail core, it will enter into the cell wall and membrane. Then the virus injects the DNA as a syringe which infects the vaccines. Like that it will inject its nucleic acid into the bacterial cell. Coming to the third phase, that is transcription and replication. In transcription and replication, how it will happen, let us see. So bacterial mRNA, that is RNA produced from the bacterial chromosomal DNA, and bacterial proteins, so they used to stop synthesizing immediately after the phage infection. That is, whenever the phage DNA enter into the bacterial cell, the bacterial mRNA and production of these bacterial proteins, they used to be stopped. And bacterial DNA also used to be degraded into small fragments, and that nuclear region of the bacterium also becomes dispersed throughout the bacterial cell. Why it will happen means, this virus, it will make to synthesize the different enzymes by the bacteria which will degrade this DNA. Let us see how it will do in the next step. So some phage mRNA is also made immediately after infection. Then here bacterial mRNA is degraded and its synthesis also stopped, but in contrary the phage mRNA synthesizing more. and amount of phage DNA also increases after a brief delay. Then specific proteins used to be synthesized from the this DNA and RNA which are produced from the phage, followed by appearance of organized capsid precursors. That is, from phage DNA, RNA is synthesized, that is mRNA is synthesized. From mRNA the proteins are synthesized through the transcription. Then these proteins form initially that precursors of this encapsid protein used to be synthesized. From these precursors, that capsid protein used to be synthesized in the bacterial cell. These capsid proteins, they will enclose the bacterial nucleic acid. Then finally it results in the formation of mature infectious capsids. Then after formation of mature infectious capsids, then this early some genes used to be synthesized from this phages, that is early phage genes. They used to be, how they will transcribe, that is how the mRNA used to form is by using host RNA polymerase. Because of bacterial RNA polymerase, the DNA is transcribed into the mRNA, then mRNA translated into the protein. So these genes code for the nucleases which will break down the host DNA, and some delayed early genes, that is already early genes. What early genes? They use the bacterial RNA polymerase to synthesize the mRNA, and delayed early genes, they used to code for the phage enzymes which produce unique phage DNA constituents like 5-hydroxymethyl cytosine. This 5-hydroxymethyl cytosine, what it will do? It will replace the cytosine in the bacterial DNA. So this compound, it used to replaces the cytosine present in the bacterial DNA. And some delayed genes also code for the polymerases and ligases which play very specific role in the phage DNA replication and recombination. And finally late gene products are there. That late gene products also include some structural components of that new phage particles, and lysozyme used to be synthesized. That lysozyme will lyse the bacterial cell and releasing the mature virions. And coming to the fourth step, that is assembly and release, that is the final step. After synthesis of this mature encapsulated bacteriophage particles, that is structural proteins and nucleic acid, phage components begin to assemble into a mature phages. After assembling of mature phages, that is 25 minutes of the initial infection, these bacteriophages release out of the host bacteria by lysis to infect the new bacterium. That's why this phase is known as lytic phase. There will be another thing like lysis inhibition. Lysis inhibition means when in the host cell, that is extra environment outside the host cell, if it is full of lytic phages, then the release of this fully developed bacteriophages from the host cell, bacterial cell, will be inhibited. That is known as lysis inhibition. That is, some lytic phages undergo a phenomenon known as lysis inhibition, where completed phage progeny, they will not immediately lyse out of the cell if extracellular phage concentrations are very high. That process is known as lysis inhibition. Coming to the lysogenic cycle, as already discussed, in lysogeny, the viral DNA of the temperate phage, instead of taking over the functions of the host cell genes, it used to incorporate its host DNA and becomes a prophage in the bacterial chromosome, acting as a gene. What is this prophage? Prophage is a stage where integration of the viral nucleic acid into the bacterial chromosome takes place. That stage is known as the prophage. In this, the bacterial cell metabolites, here the bacterial functions are not disrupted. Bacterial cell normally metabolizes and reproduces normally, and the viral DNA is also transmitted from host, that is mother bacterium, to each daughter cell in a successive generations. Sometimes what it will happen, the viral DNA is removed from the host chromosome and the lytic cycle occurs. That stage is known as spontaneous induction. That is how it will happen. A change from lysogeny to lysis, how it will be induced, either through irradiation with ultraviolet light or by exposure to some chemicals. This phase will happen. So now let us discuss about the differences between the lytic cycle and lysogenic cycle. In lytic cycle, what it is going to happen, it is a type of viral reproduction mechanism which results in the lysis of the infected cell, whereas lysogenic is a viral reproduction mechanism where viral DNA is integrated into the host genome. And in lytic cycle, viral DNA is not integrated into the host genome, whereas in lysogenic cycle, viral DNA is integrated into the host genome. In lytic cycle, host DNA is hydrolyzed, that is 3-methyl cytosine hydroxycytosine, which will replace the cytosine of the bacterial DNA, that is host DNA is hydrolyzed. But here nothing will happen like that. And in lytic phase, it does not have any prophage stage, that is integration of the viral nucleic acid into the bacterial chromosome, but lysogenic cycle have the that prophage stage. And in case of lytic cycle, DNA replication occurs independently of the host DNA replication mechanism, because it is degrading the host DNA and it is starting synthesizing its own materials. But in case of lysogenic cycle, viral DNA replication takes place along with host DNA replication mechanism. And here in lytic cycle, host cell is lysed as the viral particles are released from the host cell. Here host cell is not lysed. This is the total difference between lytic cycle versus lysogenic cycle. So today we understood about the lytic cycle and lysogenic cycle, that is the bacteriophage infecting the bacterial cell and how they will behave in the bacterial cell after infection. So this is completely about the lytic and lysogenic cycles, and thank you for your keen interest in this video, and remember to like and subscribe for more videos. And if you feel this is helpful video, please share with your friends. If you will subscribe to my channel, then it will motivate me to keep more videos. I wish you great health, prosperity and happiness. Thank you one and all.